The overall goal of this procedure is to demonstrate how to implement time-gated fluorescence lifetime imaging microscopy, or FLIM, in an automated multi-well plate workflow using open-source software and basic instrumentation.
This methodology can be applied to FLIM-based readouts in assays of fixed or live cells and is particularly useful for reading out cell signal processing of protein interactions. The main advantages of this technique are that FLIM provides robust quantitative readouts and can provide the interaction population fraction in a single measurement.
Applying global analysis techniques to thousands of cells over hundreds of fields of view lets us utilise forced-to-resin energy transfer, or FRET, readouts, and taking advantage of fluorescence lifetime changes down to about 20 picoseconds. Demonstrating the procedure with Frederik Görlitz will be Sunil Kumar, our research associate, and Ian Munro, our software developer.
Begin by starting the Micromanager and open the OpenFLIM HCA plugin. Under the FLIM Control tab, select Delay Box Calibration File and open the Calibration Files. Select the calibration file for the specific combination of the delay generator unit and laser repetition rate of interest. Under the File menu, select Set Base Folder and select the folder where the data will be saved. After confirming that all the appropriate parameters have been set, manually set a gate width of 4 nanoseconds at the gated optical intensifier control box for the whole experiment. To acquire instrument response function image data, manually place a